Okay, so this will be the second video that I'm recording myself, right? You will have seen a lot of other videos from YouTube. So in fact, there are a lot of resources out there, right, for the process of meiosis. Not all of them are that good, right? So for those that I've chosen, I hope you find them useful. But I thought those are quite good lah, and at least no mistakes, right, in their videos. Okay, so anyway, this is the video that's going to talk about the changes in DNA content and chromosome number during the process of meiosis. Okay, so before we look at the changes, right, we need to be sure how do we count, right, the number of DNA molecules and the number of chromosomes, right, so that we can then check the changes. Okay, you should have done this in mitosis already, but if you were not sure already, after mitosis, then just another round here lah, right, to look at all these changes. Okay, so anyway, right, looking at this figure, which you also have in your notes, okay, the figure on the left, you can see 2 chromosomes, right, and each chromosome contains only 1 chromatid, right, because this is before DNA replication, right? So each chromosome would be made up of 1 DNA molecules, so 2 chromosomes, 2 DNA molecules. Okay. After DNA replication, right, the number of chromosomes is still 2, right? The 2 sister chromatids, okay, or rather, DNA replication would have formed, right, 2 sister chromatids from each of the chromosome earlier. Okay, and the 2 sister chromatids are joined at the centromere, right? So we still count that as 1 chromosome because there's only 1 centromere. Okay, so here we have 2 chromosomes, each with 2 sister chromatids. Each chromatid is 1 DNA molecule, right? So therefore, right, each chromosome has 2 sister chromatids. Each chromosome has 2 DNA molecules, 2 chromosomes, so 4 DNA molecules in total. Okay, so once you know, or once you are sure about this already, right, then it is much easier for you to track, right, the changes in the amount of DNA and the chromosome number, right, both for mitosis and meiosis. Okay, but anyway, right, here I'm putting the amount of DNA as the number of DNA molecules. Okay, however, right, in quite a number of questions, they don't really use the number of DNA molecules as the unit, right? They will just represent the amount of DNA with some arbitrary unit. Okay, so instead of 2 DNA molecules, maybe they will just call it 2x, and therefore 4 molecules of or 4 DNA molecules would be 4x. Right, so the arbitrary unit, it depends on what they give you originally lah. So you just tackle it accordingly. All right. So anyway, let's move on to this graph, which you also have in your notes. Okay, so I'll be focusing on meiosis. Okay. So in the original cell, right before the whole cell cycle, right, there is 2x amount of DNA. Okay, then the amount of DNA per cell increased, so that must be S phase during the interphase, right before meiosis. So S phase will have doubled the amount of DNA from 2x to 4x. The graph is drawn with a slope because DNA replication obviously is a continuous process, right? So the amount of DNA increases continuously until the end of DNA replication in S phase. Okay, then we have the drop here, right, from 4x to 2x, and you need to take note of the y-axis when you are looking at such graphs, right? It says DNA content per cell, right? So when it says per cell, then obviously the part where the DNA, the amount of DNA drops would be during cell division or cytokinesis, right? So this is the first cytokinesis after meiosis 1. Sometimes I just call it cytokinesis 1 for short, right? So that makes this part, this portion here, meiosis 1. Of course, includes a bit of G2, right, and then the meiosis 1, yeah? So if the y-axis is per nucleus instead of per cell, then this would be telophase 1 instead of cytokinesis 1. All right. So anyway, let's move on. Okay. So we have a second job here. So that obviously, right, would be our cytokinesis 2. Okay. So that makes this part meiosis 2. Right. So after this part, right, the cytokinesis 2, then obviously all these are gametes. Gametes have half the amount of DNA as the original, so therefore, right, 1x instead of 2x. Okay, then the amount of DNA per cell goes back to 2x, so that must be fertilization, the fusion of 2 gametes. So this would be the zygote, right, with the normal amount of DNA, 2x. Okay. Yeah, so this is how you read such graph, right? And if you understand how to read the graph and you understand the process of meiosis, you can also draw the graph yourself. Okay, so we'll move on to some think time questions that I've included in the notes. Okay, so if you haven't done this already, right, spend some time looking at the diagram, recalling the process of meiosis also, and fill up the questions yourself first before you continue with the video. So you can pause the video, yeah, before or after which you can then resume to check your answer. All right. Okay, so let me continue. Okay, so the original cell, right, is given that the DNA content is 2x, and you can see there are 2 chromosomes in this cell, right? Then after S phase, after DNA replication, there will be double the amount of DNA, but the number of chromosomes is still 2x. As we mentioned at the start of this video, right, the 2 sister chromatids are joined together at the centromere, so counted as 1 chromosome, so 1, 2. So the 2 homologous chromosomes, right, will form a bivalent in prophase 1. That is still counted as 2 separate chromosomes. So it's 1 bivalent, but 2 chromosomes, or the pair of homologous chromosomes. The amount of DNA obviously doesn't change, so that is still 4x. Okay. Then after anaphase, when the homologous chromosomes are separated, there will still be 4x amount of DNA, no change, and still 2 chromosomes. So whether they are joined together as a bivalent or separate as individual homologous chromosomes, is 2 chromosomes now, right? No difference. Then we would have finished the cytokinesis 1, and this is prophase 2, one of the daughter cells at the end of cytokinesis 1. So because of that, right, cell division has occurred. So the amount of DNA per cell would have halved. Right, so instead of 4x, that's only 2x, and the number of chromosomes, as you can see here, only 1. Okay, then after anaphase 2, when the sister chromatids are separated, right, the amount of DNA doesn't change, but the number of chromosomes would double. Why? Because at first it's 1 chromosome with 2 sister chromatids joined together. Now the sister chromatids are separated, so each chromatid is counted as 1 chromosome on its own. Right, so therefore 2 chromosomes here. But no change to the amount of DNA because it's still the same amount, as you can see in the diagram, yeah? Then there will have been your telophase 2 and cytokinesis 2 to form the gamete. So for each gamete, right, there's only 1 chromosome and X amount of DNA, right, which if you compare back to the original cell would be halved for both the DNA content and the number of chromosomes. Right, okay? Okay, then there will be a second question, right? Given this cell with 3 pairs of homologous chromosomes, or 2n equals 6, what would be the changes in the number of chromosomes per cell during the whole of meiotic cell cycle? So G1, no change, 6 chromosomes in total. S phase, there's double the amount of DNA, but the number of chromosomes stays the same as we have discussed earlier. No change in G2, still 6 chromosomes. Okay, and then in meiosis 1, no change, because whether it is prophase 1, the 3 pairs becomes 3 bivalents, or anaphase 1, when the 3 bivalents separate back into 3 pairs of homologous chromosomes, right? And then telophase 1 to form 2 nuclei, right? The amount of or the number of chromosomes in the same cell per cell, right, is still the same, right? Still 6. Then cytokinesis here on this graph is given as a process with a duration, right? So during the process of cytokinesis, the number of chromosomes per cell stays the same all the way till the moment when the cell has separated, right? So imagine this is an animal cell, right? You have the cleavage furrow. The cleavage furrow will increasingly close up, right? But it is still one cell until the moment the cleavage furrow has fused, and then it forms 2 cells. So it's one moment, right? It's not a continuous process. That's why it goes straight down at the end of cytokinesis. Understand that? Okay. So at the end of cytokinesis, when at that moment 2 daughter cells are formed, each cell would only have 1 set of chromosomes, and 1 set is 3. Right, so 3 chromosomes drop all the way to 3. Okay, then in meiosis 2, it will stay at 3 all the way till anaphase 2, when the sister chromatids separate, right? And each sister chromatid is counted as 1 chromosome, so the number of chromosomes double at the end of anaphase 2. Then it stays the same in telophase 2, right, until cytokinesis, or the end of cytokinesis, where the daughter cells, the gametes are formed, and each would have only 3, right, chromosomes. Yep, and it stays at 3 until fertilization. All right, so this is the graph showing you the changes in the number of chromosomes per cell.